Hello everyone. Very good evening, everyone. How are you all? I hope you're all doing fine. So we are almost in the end stage of our workshop. Mainly three lectures are left. If we are finished today, then maybe by Thursday we are done. So maybe two lectures will be left. So today I want to take care with the fluorescent analyst of your cell sorted analysis. So will not take much time. I will start the lecture slowly students will join in. So let's say we wait for 2 more minutes and then we start. So first in this part we will discuss the various fundamentals and applications of fluorescence activated cell sorting, that is FACS, and how FACS analysis is done. Here we can this machine by the BT Bioscience is a small machine, and mostly in one of the all universities or in most of the diagnostic labs this machine is always there, FACS analysis. So in this topic, we will take care of the basics of flow cytometry, understand a flow cytometry sorter works and how cells are sorted, and description of different cell sorting possibilities, and tips on sample preparation and overview of sorting services provided by the FCF. So first, which technique is the best? So if you have a sample, they could be of two types: fluid particles in suspension or tissue. Within the fluids, which are particles in suspension, could be analyzed by three methods, that is fluorescence activated cell sorting FACS analysis that we're going to discuss today. There is also microfluidic large particles also, which is COPAS. It's much advanced version of the FACS analysis. And if you have a big tissue, then you can do the laser micro dissection microscopy. It's even the more detail to understand the various cells, which kind of cells the tissues has. It's even more advanced. So these four methods are being used, but today our focus is on this FACS analysis. So what is flow cytometry? It's basically flow means something like fluid and cyto means cells and metry means measurement. So we are basically measuring the fluidic cells, the cells which are in the flow. We are measuring how many cells are present within the medium. So there are types of particles that could be analyzed. Your monocytes, WBCs, all the cells with the immunological studies. Chromosomes could be studies, bacteria, yeast, microparticles like beads. So we can study all different sort of various particles or cells in order to analyze them. So what particle size we can analyze? we can analyze from micrometer to even a nanometer. So if a bacteria is size of 0.5 micrometer, the smallest, so it could go to the monocytes of 14 micrometer. So it can range to any range it could find out. Beyond that, what does it measure? It's majorly measuring is the relative size, how much sizes of cell is present, how much relative internal complexity is there, and a relative fluorescence intensity, how much relative fluorescence it's being provided there. And what kind of samples we can run in this? So we can run mainly, we can have these analyzers, 6 different analyzers, each having different prices and have different, their own capabilities to do so. So this one is the most like cheapest one, this analyzer, and mostly being sent to the all like for the basic learning about FACS analysis. But if you want to have your own commercialized one, so these sorters are the best, these last two ones. So sorter could be divided into 3 main subsystems: fluidic, optics, and electronics. So FACS analysis machine could be divided mainly into 3 parts, that is fluidics, optics, and electronics. In the fluidics, we have to introduce the focus and then the cells of interrogation that we want to, and create a stable break-off for sorting. And in the optics, we have to generate and collect the light signals, and in the electronics, to convert the optical signals to proportional digital signals and process the signals and communicate with the computer. So how can we record individual cells? Yeah, so the sample is introduced into the running sheath, into the flow cells, and sample is hydrodynamically focused in the core stream, and sheath fluid and samples do not mix in it. Yeah, so there is a sheath fluid is going on, and there is a central core is there, and single flow is flowing it. Yeah, so here in this single flow we measure the things actually. So here the sheath fluid and the single flow, they do not mix with each other, and reduction of cross section causes an acceleration of the central core stream that leads to laminar fluidics, and sample flow adjustable is taken care. So there are 2 different optic system in a sorter. So then now comes the like optic systems. It could be consist of laser and fiber optic cables that carry beams to the steering prisms and direct laser beams to the stream. So here we are having various lasers, 1, 2, 3, 4, 6 kinds of lasers, and they're all providing their power, and with the various prisms they're being focused and then put in over the flow cells. Our cells are being flow here, the stream of cells, and then they are being passed over it. So here we can see the results later on. So there are 2 different optic systems in a sorter. Collection system consists of fiber optics that directly emit the light to the appropriate emission filter block, and the filter that directs the signals in the emission block to the appropriate photomultiplier tube, that is PMT. So this is the filters that are arranged. It could be in trigon form or in the octagon form. Yeah, there could be 3 filters here, A, B, C, or 8 filters here all together. So in any form it could be present. And what is this PMT is doing? It's actually photon of light is being there, giving these electrons, and they are being focused by the diodes in the vacuum tube. So these diodes, they're doing, they're just getting reflected back and again. They're being reflected, reflection, reflection, reflection. So by the end, you have a lot of these particles of electrons which are carried over the anode. So which could be used for further current produced in this FACS analysis. So how cells are actually sorted in this? So first, here our sample generates light scatter fluorescent signals. Signals are analyzed here. Yeah. And then charge is applied via stream changing via the second. Then the charge droplets break off. Then the charged particles of cells, they started to getting separated of the charge. The light red and green dots we can see. Then there is a magnetic fields here, which according to that magnetic field, your charged particles will deflect it towards that part. The red will go to the left, green will go to the right. So in the center, you will have the neutral ones, the uncharged particles, and they will go to the waste. And in this way, we can sort out our other red particles and green particles into the different collection tubes. This is interrogation point where the break off is taken place, and the green part is being distributed here and the orange part being distributed here. So live image of the stream taken from the video monitor. So this is the single drop is going on and your drops are getting separated like that in the analyzer. So what kind of devices we can use in this? So this is the machine that looks like the magnetics. We can see these two magnetics here and the cell sorter here on the top, and your droplet is coming down here and your cells getting separated in these two different tubes inside that. This is the inside of your this machine. So you can sort your cells in 1.5 ml tubes, FACS tubes, beaker, anything that you feel, yeah, or you can use 96 well plate, go for 8 well plate, 6 well plate, so whichever you feel like, go with that. Some quick tips for making these samples. So before planning your experiments, bring your controls, yeah, so unstained and non-transfected cells to estimated background signal, bring single stain samples to set compensation, bring FMOs if it sets gates. So mainly 2% of your fetal calf serum has to be there, not more than that. If you do it, so cell might be sticky and might be not sorted out. Don't use phenol red media because that it will also give autofluorescence, and most cell types do well in just PBS and FCS. Beyond that, in order to have better results, use calcium magnesium free sorting buffer or add EDTA to that. Or beyond that, you can do your cell lysis with 25 microgram per ml of DNase 1 and 5 millimolar magnesium chloride, and use 1% aquatase in the sorting buffer. So all samples has to be filtered directly before sorting. So there are some nozzles coming up for 70 micrometer nozzle, 85 micrometer, 100 micrometer. So you can use any size of them. In summary, if you can measure in this game what we have discussed just now, what kind of sorter you want to do it, you can sort between smallest bacteria to largest adipocytes. Plan your fluorochrome panel which you want to use it for the sorting. Make sure that you want to do bulk or single sorting. Don't forget to use controls, unstained, single stains also, and bring appropriate media for downstream techniques. So that was a simple little bit touch about what is this fluorescence assisted sortings looks like, giving you a brief overview about it. Yeah, July 15th is final, or you can take 17th or 18th is also fine with me, no worries. So let's see one like 3D like insight cytometry FACS analysis. So let's have a look over this.
Hello guys, in this video I'm going to speak about a very important or very interesting mechanism called the flow cytometer and FACS. Actually I did two videos, and in this video I'm going to mention specifically flow cytometer, and in the next video I'm going to mention the FACS mechanism, fluorescence activated cell sorting. Let's start speaking about flow cytometer. Flow cytometer is a cell counting mechanism. So it's used to count the cells and to analyze the phenotype and the health situation of the cell. How does it work in general? First, in general, in flow cytometer, I have something called the flow cell. In the flow cell, we add our cell mixture. Then I have the sheath fluids. The sheath fluids will apply something called the hydrodynamic focusing. The hydrodynamic focusing of the sheath fluids will send the cells one by one through the flow cytometer tube. It's very important to to pass the cells one by one through the tube, because here I have something called the laser beam, and the cells should pass through the laser beam one by one. This point where the cells pass through the laser beam is called the interrogation point. Now, when the cell passes through the laser beam, there are two events happening. First is the forward scattering, which is the light scattered in an acute angle to the forward direction, and the side scattering, which is the light scattered in every other direction, in side directions. These two events, or this light scattering, is detected by specific detectors called the forward detector and the side detectors. And these detectors are connected to a PC or a data analysis analyzer, which is going to analyze the data obtained from the detectors. Now, this is in general. Now I'm going to speak more in details. Saying that this is the flow cytometry tube, and then the cells pass one by one through the laser beam. When the cell pass through the laser beam, the cells scatter the light in a forward direction, which is the light scattered in an acute angle. This forward scattered light is directly proportional to the size of the cell. Why? Because the cells scatter as like a particular amount of light in the forward direction, but when the cell is bigger, the forward scattered light will be larger, or like more forward scattered light will be detected. And when the cell is smaller, the cell will scatter less light in the forward direction. So the forward scattered light is directly proportional to the size of the cell. Now the detector detects the pulse of photons, which are the forward scattered light is a pulse of photons which are detected by the detector. And the detector then is, as I told you before, is connected to a PC or a computer. The PC will convert the intensity of the light detected by the detector into voltage. And then I will get a specific amount of voltage for every cell passing through the laser beam. The PC will give me a data similar to this. So every time a cell passes through the laser beam, I will get a specific peak, like high voltage peak for large cells and low voltage peaks for small cells. The data, or the forward scattered light data at the end, will be similar to this. Time versus voltage, and then the peak will be proportional to the size of the cell. This is forward scattered light. The second event happening in the flow cytometer is the side scattered light. So when a cell passes through the laser beam, the cell will scatter the light in side direction. And this side scattered light is directly proportional to the complexity and granularity of the cell. Why? Because the simple cells scatter the light in the side direction, yes, but when we have granules inside the cell, so when the cell is complex from inside, these little granules will also scatter light in different directions, and in this case I will get more side scattered light, like this. So this is a complex cell from inside. It contains small granules inside, and then these granules will also scatter the light in different directions, and in this case I will get higher intensity of the side scattered light. Similarly to what we saw in forward scattered light, the detectors are connected to the PC, which is going to convert the intensity of light into voltage, and then I'll get something similar to this. So the side scattered light data will be similar to the forward scattered light data. So for the complex cells will give me a high peak, and the simple cells will give me a small peak. Now, in order to analyze the cells, what I have to do is that I can combine both forward scattered light and side scattered light data together. In this case, I'll get something called the two-dimensional histogram. The two-dimensional histogram, from its name, it's two-dimensional. So I have side scatter and forward scatter data all together. In this case, what I get is something similar to this. So I have here 4 quadrants. If the cell appear in the lower quadrants, it means that it has low signal on the side scatter detectors, which means that the cell is simple. If the cell appears in the upper half, it means that it gives high signal on the side scatter detectors, so it means that the cell is complex. If the cell appear on the right half, it means that the cell gives high signal on the forward scatter, which means that the cell is big. And if the cell appears here, it means that the cell is small. So if the cell appears, for example, in this quadrant, it means that the cell is small and it's simple. If the cell appears here, it means that the cell is small but it's complex. Here, it means that the cell is big and complex, and here, it means that the cell is big but simple. I will give you a very good example of this, which I have an example about the analysis of a sample of white blood cells. So what we see here is an analysis of white blood cells. This is side scatter and this is forward scatter light. What we see here is a group of cells which are complex, very complex cells, because they give high signal on the side scatter detector, and they are medium inside. They are not so big and they are not small, so they are medium inside. Obviously, these cells are granulocytes, because granulocytes, or like eosinophil, basophil, neutrophils, they are medium inside, but they are very complex cells. Here we have bigger cells, because they give almost like they give more or less high signal on the forward scatter, but they give low signal on the side scatter. So they are big, but they are not complex. They are simple cells, and these are monocytes, because monocytes are big cells, but they are not complex. Here we have very simple cells, because they give very low signal on the side scatter, and they are like smaller than monocytes and granulocytes. So obviously these are lymphocytes, because the lymphocytes are smaller in size and they are simple cells. Of course, this is a very useful technique used to count the cells and to analyze the phenotype and the health situation of the cells. And what's very special about this technique is that we can analyze many thousands of particles and cells per second. It's like it's an easy technique. It's not very hard to perform, and it's precise. It's very used nowadays in all the laboratories. This is everything I wanted to tell you about flow cytometer, which is very important to know if you want to know more about FACS mechanism. In the next video, I will speak about FACS mechanism. So if you want to know more about FACS mechanism, then you need to watch the next video. If you like this video.
So basically in this, so your eosinophil, basophil, neutrophils are being separated, monocytes on this histogram, and lymphocyte on this one. So everything is sorted out with the forward angle scatter and the side scatter. Now it's time to even further scatter them according to their complexity. We will discuss them in the next part.
Hello guys, this is a continuation of the subject flow cytometer and FACS mechanism. In the previous video, I spoke in details about flow cytometer, and in this video I'm going to speak speak about FACS mechanism, fluorescence activated cell sorting. If you didn't watch the previous video, I highly recommend you to go and watch it, because it will be much easier to understand FACS mechanism if you fully understand the flow cytometer mechanism. So let's start speaking about FACS. FACS mechanism is not only a cell counting mechanism, but it's also a cell sorting mechanism. So it provides a method to sort a heterogeneous mixture of biological cells. It depends on light scattering, as we saw in flow cytometer, so forward scattering in light and side scattering light, but it also depends on the fluorescence, and it also can detect fluorescence. FACS mechanism looks like this. So we have the machine, we have of course the computer, which is going to analyze the data, and we have the fluids container and the waste container. Now, how does it work? A FACS machine looks like this from inside. So we have also the flow cell and the sheath fluids, which apply the hydrodynamic forces on the cell to pass them one by one through the tube. This is what we saw in flow cytometer in the previous video. The cells will pass one by one through the cell tube, and then we have the laser. So the cells will pass through the laser beam one by one. When the cell pass through the laser beam, there is the forward scatter, which is detected by the forward scatter detector, and then there is the side scatter, which is detected by the side scatter detector. But here there is also the fluorescence, which is detected by the fluorescence detectors. Here we have mirrors and filters, which are aimed to filter the light and to deliver particular wavelengths to the appropriate detector. Then all the detectors are connected to the computer, which is going to analyze the data. Now let's speak more in details. As we saw in in flow cytometer, we have forward scatter and light and side scatter. Here we have also the fluorescence. So saying that I have a cell mixture. Inside this mixture, I have a mixture of cells, or cells who are expressing a particular protein, and other cells who are not expressing the particular proteins, the particular protein or the protein of interest. What I want to do here is I want to count how many cells there is inside the mixture, and how many cells are expressing the protein and how many cells are not expressing the protein. And then I want to separate this heterogeneous mixture into two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. To do this, first of all, I have to design a specific antibody for this protein. This antibody is specific to the protein of my interest. I'm not like I can buy this protein. There are companies who design specific antibodies for particular proteins. Then I have to label this antibody with a specific fluorophore. Now what I have to do is that I have to add this antibody, labeled antibody, to the mixture of cells I have. When the antibody is added to the cells, this antibody is going to bind specifically to the protein of my interest, and then I will have labeled cells. So I will have the cells who are expressing the protein will be labeled with this fluorophore. Now, when the cell passes through the laser beam, this is going to happen. First, I will get the forward scattered light, which is detected by the forward scatter detector, and this is proportional to the size, as we saw in the previous. We saw this in details in the previous video. So forward scattered light is directly proportional to the size of the cell. Side scattered light is directly proportional to the complexity of the cell. And then the fluorescence will give me an information about which one of the cells is expressing the protein and which one of the cells is not expressing the protein. Now when the cell passes through pass through the laser beam, this is the laser beam. First of all, this is the forward scattered, which is detected by the forward scatter detector. The side scatter, which is detected by the side scatter detectors. And then when the fluorophore pass through the laser beam, the fluorophore will be excited, and then when the fluorophore is excited, the fluorophore will emit light at a particular wavelength, and then this light will be detected by the fluorophore detectors. All these detectors are connected to the computer, which is going to give me the exact data about every event of these three. Now, of course, when the cell is expressing more proteins, there will be more fluorescence coming out of this cell. At the end, I will get a data similar to this. So when the cell is expressing the protein, I will get a high peak, and then when the cell is not expressing the protein, I will get a small peak. And then the computer will give me something called the one-dimensional histogram, which is something similar to this. Here we have the cell count, or the events, we call them the events passing through the laser beam. And here we have the signal of the fluorophore. So when I have a high signal of fluorophore, then these cells are the cells who are expressing the protein of interest, and the cells who give low signal on the fluorescence detectors are the cells who are not expressing the protein. What I can do further is to combine this data with other data I have. So for example, I can combine the data obtained from the fluorescence detectors with the data obtained from the forward scatter detectors, for example. So here I have the fluorophore, here I have the forward scatter, or here I have the fluorescence and forward scatter light. When the cell appears here, it means that these cells are small cells because they have low signal on the forward scatter and they have low signal on the fluorescence detectors, so they are not expressing the protein. When the cell appears here, it means that the cell is not expressing the protein because it has low fluorescence, but it's a big cell because it has high signal on the forward scatter light. Then when the cell appears here, it means that it's expressing the protein, but it's a small cell. And when the cell appears on the upper right quadrant, it means that the cell is big and it's expressing the protein. So this is what we call the two-dimensional histogram. I can combine two data together and then I get something like this. Now, what's special about FACS mechanism, or why it's called FACS? FACS means fluorescence activated cell sorting, which means that this mechanism cannot only give me a cell count, cannot give me a data about the cell count or the percentage of the cell expressing the protein and not expressing the protein, but also the FACS can separate the cell mixture into two groups of cells. How does it work? Let's see. This is the FACS mechanism. So here we have the flow cell, and here we have the cells passing one by one through the laser beam. When the cells pass one by one through the laser beam, as we saw before, it will scatter light forward and side scatter light, and also fluorescence. All these events will be detected by the detector. At the end of the tube, there is something called drop formation. In drop formation, we have a mechanism called a vibrating mechanism. This vibrating mechanism will separate my mixture into small droplets. These droplets should contain only one cell by droplet. This is very important. Then what I can do as a user is that I can adjust the settings of the program in order to charge the cells. So at the end of the tube, I have something called the electrical charging ring. The electrical charging ring is responsible to charge the cells according to my adjustment. So, for example, if I adjust the settings, for example, of the program, I can ask the computer to charge the high fluorescence cells with a positive charge and the low fluorescence cells with a negative charge. Then the electrical charging ring will charge the cells will pass through the laser beam. The detector will detect the fluorescence, and then the computer will charge the high fluorescent cell with positive charge, for example, and the low fluorescent cells with negative charge. I can choose, actually. For example, if I have this data, this is the two-dimensional histogram, I can choose to charge the big high fluorescent cells with a positive charge, and the big low fluorescent cells with a negative charge, and the other cells with no charge. Then the neutral cells will go to the waste. These cells might be like debris. These might be like just debris. I'm not interested in them, so it depends what cells I'm interested in. So I can adjust the settings of the program, and then the electrical charging ring will charge the cells. Then I have different electrodes, like negative and positive electrodes, and then the positive cells will go to the negative electrodes, and the negative cells will go to the positive electrodes, and the neutral cells will go to the waste. In this case, I will have two groups of cells. The cells who are expressing the protein and the cells who are not expressing the protein. And this is what we call cell sorting. Of course, I can also study two proteins, not only one. And this is done by using different... of course, to study two proteins, I need two specific antibodies, because I need one specific antibody for each protein. But I need to label every antibody with a different fluorophore. Of course, I have different fluorophores, and every fluorophore emits light in a different wavelength. I need to know every fluorophore, the wavelength every fluorophore emits light in. So if I use two different fluorophores, then I can study two different proteins. For example, this fluorophore emits a green light or a light green light, and this fluorophore emits yellow light. So they are different wavelengths. Then if this cell is emitting only has only a green fluorophore, we can know that this cell is expressing one protein, or the green protein. If the cell is emitting yellow light, then the cell is expressing only the second protein. And if the cell is emitting two lights, or giving two signals on the two detectors, then we can say that the cell is expressing both proteins. How can we detect two different wavelengths? As I showed you before, the FACS mechanism contains different detectors, different fluorescence detectors, and here we have the filters, the light filters, which deliver a particular wavelength to a particular detector. So every detector will detect a particular wavelength, and then we can use two different fluorophores if they emit light on different wavelengths. Okay. We can also combine the data together. So I can form this two-dimensional histogram again, but with the protein 1 and protein 2. So I have fluorophore 1 signal coming from fluorophore 1 and fluorophore 2. So when the cells are in this, when the cells are here, it means that these cells are expressing both proteins, because they are giving high signal on the first detector on the second detector. If the cells are here, it means that they are expressing protein 2, but they are not expressing protein 1. These cells are expressing protein 1 because they have high signal on the detector of fluorophore 1, but they are not expressing protein 2 because they have low signal on the detector of fluorophore 2. and these cells are not expressing any of the proteins. Now I have like a real two-dimensional histogram, and this is like ideal to see how cells are shown in the two-dimensional histogram. So these cells, here we are studying two different proteins, CD14 and CD4. We are labeling CD14 with PE, and we are labeling CD4 with FETC. We are labeling them with different fluorophores. Then cells in this quadrum are expressing CD14, but they are not expressing CD4, so we have CD4 negative. Cells here are expressing both. They are giving high signal on both of them. So the CD14 positive, CD4 positive. These cells are CD14 and CD4 negative because they are giving low signal on both detectors. And these cells are expressing CD4. They are giving high signal on CD4 detector, on FETC detector, but they are giving low signal on the other detectors, so they are expressing only CD4. This is like a very simple example about how the data looks like in the two-dimensional histogram. Of course, there are many more complex data. So if you want me, I cannot speak about everything in this video. So if you want me to do another video especially about data analysis and gating in FACS analysis, please write me in the comments. As you know, now we can in the new FACS machines, we can analyze like tens of proteins and tens of cells in one time, because new FACS machines have like some machines like there is now one FACS machine in the market which contains 10 lasers and 30 detectors. Can you imagine 10 lasers and 30 detectors? So you can analyze many proteins and many cells at one time. But of course, the data then will be much more complex than this data. This is a very simple data. If you want me to talk more about this, then write me in the comments. If you enjoyed the...
So that's it. So we have discussed basically the basics behind of your FACS analysis. Now let's see one animation behind this. Small animation.
Cell processes are controlled by complex and dynamic intracellular signaling networks. Surface receptors pass information into the cell by modifying cytoplasmic proteins. These modifications include phosphorylation, methylation, and acetylation, which control a broad range of cellular functions including growth, development, and apoptosis. Disruption of signaling networks often leads to disease. To understand signaling processes in normal and pathological conditions, identification and quantification of signaling proteins is necessary. Flow cytometry is ideal for such a task. Flow cytometry is a laser-based technology widely used to phenotype cells using antibodies directed against surface markers. It can also allow simultaneous analysis of multiple intracellular targets using antibodies that detect signaling proteins, their post-translational modifications, epigenetic modifications, and downstream transcription factors. The first step of intracellular flow cytometry is fixation of the cells using formaldehyde, which immobilizes proteins and preserves transient signaling events. Cells are then permeabilized with methanol or detergent, allowing antibodies to access to the intracellular space. Optimal fixation and permeabilization conditions depend on the location and biochemistry of the target epitopes and should be carefully considered to ensure successful antigen detection, especially when staining surface and intracellular proteins simultaneously. Since multiple signaling proteins and surface antigens can be identified using fluorescently distinct antibodies, flow cytometry can readily characterize signaling profiles at the single cell level within phenotypically distinct populations. This allows the characterization of the relationship between intracellular targets and enables detection of signaling events from rare cell populations within a heterogeneous sample. Multiplex analysis of these protein targets saves time and resources while producing data-rich results, making intracellular flow cytometry an invaluable tool to unravel the complexities of cell signaling. For more information on validated products and optimized protocols for flow cytometry, visit www.cellsignal.com/flow cytometry.
So that's it for today, students. I will share these things after the lecture is over, and then you can have a look over all these presentations. So thank you very much. Today just focusing on the facts analysis. I hope you had good learning out of it, and wish you all nice evening. Take care. Bye bye.